And you'll notice that page F1, I've entitled Metabolic Reactions. We first learned the word metabolic or metabolism on page A3. It's not a new word. Metabolism or metabolic just means biochemical. So when we talk about your metabolic reactions, we're referring to your biochemical reactions inside of any living organism. Now, when we examine the biochemical or metabolic reactions inside any living organism, there are two major categories of biochemical reactions or metabolic reactions. There are catabolic and anabolic reactions. Are these new words? No. They originally appeared on page A3. If you don't believe me, look at page A3. When we were talking about the characteristics of living things, we said living things carry on catabolic reactions to produce energy and anabolic reactions for growth. So we talked about those were characteristics of living things on page A3. Let me review this by focusing on anabolic reactions first. Anabolic means growth. It is associated with growing in size. And how living cells and living organisms grow in size is by synthesizing, by joining smaller molecules together to form larger, more complex organic molecules. Now, an example of an anabolic reaction or an anabolic process that we've learned about many, many times in section D are dehydration synthesis reactions. We first learned that on page D1, and we've spoken of it many times before. Dehydration synthesis. Synthesis means to join together. Dehydrate means to remove water. The way that many smaller organic molecules are joined together to form a larger, more complex one is by removing water. So this is the way simple sugars are snapped together to form disaccharides or polysaccharides by removing water through a dehydration synthesis reaction. We covered that. This is the way fatty acids, 3 fatty acids, are joined to a glycerol molecule to form a triglyceride. That's a dehydration synthesis reaction. It is an anabolic or growth process. It is the way that individual amino acids are snapped together to form a polypeptide chain, a chain of a lot of amino acids joined together by a dehydration synthesis reaction. It is the way that nucleotides are joined together to form large nucleic acids. So a very common way that smaller organic molecules are joined together to form larger, more complex ones is by removing water, dehydration synthesis reaction, and these entail growth, growing larger by accumulating these large, complex organic molecules. Now, another type of anabolic reaction that I have not specifically and explicitly spoken of, but I have alluded to it, including on a video, are what are called reduction reactions. Now, a reduction reaction involves when an organic molecule, when a molecule gains hydrogen atoms and electrons. So when a molecule gains hydrogen atoms and electrons, that's called a reduction reaction. Now this term, reduction reaction, always bothers students because students think, well, if you're reducing, well, how could you be gaining something? But we're using this word reduction reaction in a technical way, not the usual way that we use it in English, but it is a term that's used in chemistry and biochemistry and in biology. So a reduction reaction refers to gaining hydrogen atoms and electrons and gaining energy. The context where I alluded to this, without explicitly calling it a reduction reaction, was when we explained how hydrogenated vegetable oils are made. And so we did that back on page D10. So let's just remind you of that back on page D10. So back on page D10, this was quite a while ago. We learned that there were different types of fats or fatty acids. We learned that the fats found in animals are saturated fats, whereas the fats that are found in plants are usually unsaturated fats. And we explained how you can make, you can take vegetable oils, which are unsaturated fats, and turn them into margarine. Margarine, of course, is something that looks like butter, tastes like butter, and basically it was made from vegetable oils that were altered, making it now a saturated fat. So we explained that if you attach hydrogen atoms onto this unsaturated fat, then that makes it now a saturated fat, so now it looks and tastes like animal fats, like butter. This, when they do that, that's called a hydrogenated vegetable oil. I'm sure you've all heard that term before. You will see hydrogenated vegetable oils, also known as trans fatty acids, in many, many foods. And I spoke about this, and including on the video, where probably after you take a vegetable oil and you turn it into this altered saturated fat, yes, it now looks like butter and tastes like butter, but it's got all the same problems as butter, and it's probably even worse than butter. So it's better just, if you really like butter, just eat butter rather than eating these hydrogenated vegetable oils. But they are commonly found, and you'll hear a lot about this. Well, we just now learned that whenever you add hydrogen atoms onto a molecule, it gains calories of energy, and that's called a reduction reaction. So back on page F1, a reduction reaction involves the gaining of hydrogens and electrons and energy, calories of energy. So that's making it a more complex fat. Now the opposite of anabolic or growth reactions are catabolic reactions. Catabolic, as we originally learned back on page A3, refers to the breaking apart of organic molecules, usually for the purpose of releasing energy. And I had suggested way back on page A3, just think of the word catastrophe, catastrophic, where everything breaks apart. So catabolic reactions are breaking things apart to produce energy. So the splitting of large organic molecules into smaller, simpler ones releases energy. So when large, complex carbohydrates are broken apart into smaller and smaller molecules, that releases energy. And it's catabolic, we're breaking it apart. When you break apart triglycerides into fatty acids and then break apart the fatty acids into smaller particles, this is breaking it apart. It's a catabolic reaction that releases energy. When you break apart proteins into amino acids, when you break apart nucleic acids into nucleotides. Now, these are all examples of what we had previously called hydrolysis reactions. What does lysis mean? To break apart. And so you break these apart by adding water. We covered this originally on page D1. And hydrolysis reaction was listed right below dehydration synthesis reaction on page D1. So it's the exact opposite of the dehydration synthesis reaction, where you join molecules together by removing water in the case of a dehydration synthesis reaction. In the case of a hydrolysis reaction, you break it apart by adding water. So we had spoken of that many, many times in section D. Now another type of catabolic process, catabolic reaction, where you break apart organic molecules to release energy, are called oxidation reactions. Now we have not used that term before, but we will from now on. An oxidation reaction is ostensibly the exact opposite of a reduction reaction. An oxidation reaction involves the removal of hydrogen atoms and electrons from an organic molecule, and that releases energy. Now, we're going to be giving you a lot of examples of oxidation reactions and reduction reactions. Here's the context that some of you have heard this word before, oxidation. Some of you have gone to the drugstore and purchased products in the drugstore called antioxidants. An antioxidant is something that prevents an oxidation reaction. And we're going to talk more about this, but an oxidation reaction is the removal of hydrogens and electrons and the release of energy. So if you have an antioxidant, that prevents that from happening. Now, how do you remember an oxidation reaction is losing hydrogens and a reduction reaction is a gaining of hydrogens? So I've suggested that as a mnemonic or memory aid, you remember oil rig. It's easy to remember oil rig because if you look on the hills right behind this college campus, there's all these oil rigs right behind the hills behind us. So how does oil rig help you remember? Oxidation is a loss of hydrogen atoms. Reduction is a gain of hydrogens. So just remember oil rig to remember what oxidation reactions are and what reduction reactions are. In case there's any doubt, like do we have to know that for the test on Monday? Yes, because we're going to be talking a lot about oxidation and reduction reactions.
I thought lysis was when the cell bursts.
The lysis just means to break apart. We use that root all over the place in biology. So yes, when a cell breaks apart, that's called lysis. If it's a red blood cell, it's called hemolysis or hemolysis. But also hydrolysis is when you break apart a molecule by adding water, hydrolysis. Later we'll talk about viruses that enter cells and rupture them. They're called lytic or viruses. So that root, lysis, means to break apart, and it's used in many, many contexts. So that's why we told you what it meant a long time ago.
Doesn't oxidation happen at metals and stuff?
It happens all over the place. It happens all over in chemistry as well as biochemistry. All right, so now let's try to give a handle on this. I recognize that you talk about oxidation. You talk about anabolic and catabolic reactions, and we get a little confused. So let's try to help you understand this by looking on page F5. Page F5. And on the top left on page F5, this is what we see. A little diagram. All right, top left on page F5. So what is it showing? All of the chemical reactions that occur in any living organism are called its metabolism. Metabolism or metabolic just means biochemical reactions. We divide these biochemical reactions into two categories, anabolic and catabolic. Anabolic reactions involve growth. Catabolic reactions involve breaking apart organic molecules to release energy. So anabolic is where we form larger, more complex organic molecules. Catabolic is when we break them apart. In all living things, including in you, at this moment, you are doing both anabolic and catabolic reactions, both simultaneously. You are manufacturing large, complex organic molecules, and at the same time, you are breaking organic molecules apart. All right, so let me try to really put this together for you. Right now, as I look around the room, I observe all of us in this room are adults. And here's what I mean by adults. All of us in this room are as tall as we're going to get. However tall you are, that's how tall you're going to be. In other words, all of us in this room have reached our adult height. We're not growing any taller. There's only one direction we can still grow in. We can still grow wider. Now, on the assumption that we don't want to grow wider, right? We know we can't grow any vertically taller. So, assuming we don't want to grow wider, that means we must break down these organic molecules as fast as we make them. So however fast we are converting fatty acids into triglycerides and storing them in our fat cells, however fast we are joining sugars together to form glycogen in our liver cells, we have to be breaking down that triglycerides in our fat cells. We have to be breaking down the glycogen in our liver cells, or we will grow wider. So for me, I happen to really be in love with Chunky Monkey ice cream. So because I'm always ingesting a good source of fatty acids in Chunky Monkey ice cream, so I accumulate triglycerides in my fat cells and I grow wider. So if you don't want to grow wider, you must break down these organic molecules as fast as you make them. Does everybody follow that?
Yes.
Now, let's think about what we would have to do to actually get thinner. To get thinner, it seems that we've got to slow down these growth reactions. How can we slow down these anabolic or growth reactions? Eat less. Because the way you form these organic molecules, the way you form triglycerides and glycogen is with the nutrients that are in the foods that you eat. So if you eat less, then you've got less molecules to join together into these triglycerides and glycogen and large complex organic molecules. That makes sense? We cannot, but there's something else we can also do. Not only can we eat less, that'll slow down the growth reactions, but what if we speeded up the breakdown or catabolic reactions? Now, how can we speed up the rate at which we break down organic molecules? Workout, exercise. Because when the purpose of breaking apart organic molecules is to release energy. If you do an activity that needs energy, as you start to run, that speeds up the breakdown of these organic molecules by the cells of your body in order to produce energy to allow you to run. So if you exercise, that forces forces the speeding up of the breakdown of complex carbohydrates and fats and other organic molecules to produce energy in the form of ATP. There's our ATP word today. And so that's how it speed up the breakdown. So in other words, what I just told you, we all knew. All of us know that if you want to get thinner, eat less and exercise more. But all I've added to that is the fancy schmancy words. You want to slow down your anabolic or growth reactions, and you want to speed up your catabolic or breaking apart reactions. And that way you can get thinner. Now, incidentally, there is a difference between the type of organic molecules you accumulate. Clearly, there is a difference between accumulating triglycerides in our fat cells versus accumulating proteins in our muscle cells. It's certainly healthier to accumulate proteins in muscle cells than to accumulate triglycerides in our fat cells. But what they both have in common is you grow wider. Right? You're going to get wider. You're not growing taller. Let me ask you this question. What about a child? We've agreed that in an adult, if an adult doesn't want to grow wider, they have to break down these organic molecules as fast as they form them. What about in a child? Do we want these two categories of chemical reactions to be in balance? No. And here's why. A child is still capable of growing taller. If a child breaks down the organic molecules as fast as they make them, they stay the same height. Right? Because you just stay the same size. As adults, we want to stay the same size because we know that if we grow, we'll grow wider. But children can still grow taller. So they actually have to form organic molecules faster than they break them down. An example of this is to think about what a 16-year-old boy, 16-year-old guy, how they function. So 16-year-old guys usually are going through their growth spurt. Usually guys go through the growth spurt where they start to grow vertically taller a couple years later than girls do. Girls usually go through their vertical growth spurt around age 13, 14, 15. Guys usually a couple years later, usually 15, 16, 17. So when guys are going through that growth spurt, they basically open up a refrigerator at the house, and they are like vacuum cleaners. They literally, they will eat everything in the house. If somebody makes chicken or brings home some chicken or something, You know, everybody else takes 1 or 2 pieces of chicken. The 16-year-old guy puts on 5 pieces of chicken and then is still hungry an hour and a half later. All right? Now, they can do that because they're going through a growth spurt and they're going to grow taller. Here's what happens, though, when you reach your full vertical height. If you're still eating like you did when you were 15 or 16 or 17, if you're still eating like that when you're 20, 21, or 22, you're done growing taller.
Right.
So then that's when everybody starts to notice they start to grow wider in college. Because it was during the high school years that you were still growing taller, and if you're still eating the same way you were in high school, when you hit college, that's why everybody starts to grow wider in college, because you're done growing taller. Yes?
Now, how does this work with an obese child?
Well, because it's still, even though they're growing taller, they're still taking in more food than they're burning down, and even accounting for growth.
So
You can still, even if you're growing taller, you can still take in excessive amounts of food.
But when you were saying you don't want to...
Again, you can eat more food than you break down to allow you to grow taller, but that doesn't mean you can take in an infinite amount of food. Because you're only going to grow so tall, so fast, and if you take in more food than the rate that it's being used to grow taller, then you start to grow wider. It's just a matter of, total calories, the total calories formed versus total calories burned. Now, since we're on this page, F5, let's look right here at the right on F5. And this big glob, this thing that looks like a twisted up slinky, is an enzyme. We know that enzymes are globular-shaped proteins, proteins with a tertiary structure, a third-level structure of complexity. And we know that that coiled-up structure is actually the basis of how that protein works. We originally talked about enzymes on page D5. Now, enzymes, we learned on D5, could be argued to be the most important of all proteins in a living thing. Enzymes are proteins that catalyze biochemical reactions. There are thousands of different types of biochemical reactions occurring in the different cells of our body. We have different biochemical reactions occurring in our muscle cells, different biochemical reactions occurring in our liver cells, different biochemical reactions occurring in our brain cells, different biochemical reactions occurring in our kidney cells. And each one of these chemical reactions is catalyzed or caused to happen by a different enzyme. So if there are thousands of different kinds of chemical reactions, there must be thousands of different kinds of enzymes. Now, the way that the enzyme works is it attaches to a chemical. And the chemical that an enzyme attaches to is known as the substrate or reactant. So let's just look at this picture right here also, and this is part of the test on Monday, and enzymes. So here, diagrammatically, on the left, it shows this enzyme. In reality, the enzyme looks like this big glob, but we're representing it right here. And this enzyme is shown that it is attached to a chemical. What do we call the chemical that an enzyme attaches to? It's called the substrate or reactant, right? And that's what we wrote right here. It's called the substrate or reactant. And what this enzyme can do is it changes this substrate into something else. Here's a simple example. There is an enzyme that attaches to a substrate called sucrose. Now, you'll recognize sucrose. We originally learned it on page D1. We needed to know it for the first exam. Sucrose is good old table sugar. C and H sugar from Hawaii. That's sucrose. It is a disaccharide. So there's an enzyme that attaches to this. It's actually a digestive enzyme. It's in our digestive tract. It's produced by our cells of our intestine. And when we name enzymes, enzymes are usually named based on the substrate, the chemical they attach to, and then we put this ending, this suffix ending, ACE, A-S-E. So the name of the enzyme that attaches to sucrose is called sucrase, because it attaches to sucrose and it has this ending ase. This is a digestive enzyme. What does it do? The sucrase digestive enzyme attaches to sucrose in our digestive tract, assuming you've eaten sucrose, and we all do, and it catalyzes the splitting of sucrose into two monosaccharides, glucose and fructose. Now, this is a hydrolysis reaction. It requires water be added for this to happen, but it also requires this enzyme. So we had learned dehydration synthesis reactions are joining molecules together. Hydrolysis, what does lysis mean? To break apart. So when you break apart a molecule by adding water, that's a hydrolysis reaction. And we originally learned this on page D1. So this sucrase digestive enzyme is what breaks apart table sugar into two simple monosaccharides, glucose and fructose. And now they can be absorbed into our body. So we say that the enzyme converts the substrate into the product. Substrate is what you start with, and product is what you end up with. Now, interestingly, this is a reversible reaction. You'll notice the arrows point both this way and that way, because the very same sucrase enzyme can also attach to glucose and fructose and join them together. What determines which direction the reaction goes in is basically depends what you start with. If you start with sucrose, the enzyme breaks it apart into glucose and fructose, and that's what happens in our digestive tract. But if you start with glucose and fructose, the same enzyme can join them back together again. Of course, joining them together is called a dehydration synthesis reaction, and that's where water is removed. Now, sucrase is just one of thousands of enzymes in our body. It's, again, a digestive enzyme. Let me mention another digestive enzyme that we have alluded to previously. There is another digestive enzyme called lactase. What do you think lactase attaches to? Lactose. Now, what's lactose? What is lactose? Have we ever learned lactose? It's on page D1. We needed to know it for the first exam. It's the sugar in milk. Lacto means milk. So this digestive enzyme lactase attaches to lactose. Let's just pretend this is lactase attaching to lactose. And it breaks it apart into glucose and galactose, which are the two simple sugars that make up the disaccharide lactose. So then you can absorb glucose and galactose. You can digest the sugar that's in milk and dairy products. But some people, we've mentioned this before, as they grow older, usually after they reach around their 20s, just things start to change when you hit 20s. Some people, for genetic reasons, stop producing this digestive enzyme in their intestines. And therefore, when they drink milk and other dairy products having this milk sugar, this lactose in it, they cannot digest it. So because they cannot digest or absorb this sugar, it causes them to experience cramps and diarrhea. And we say, therefore, that they are lactose intolerant. They don't tolerate lactose. So if they want to drink some milk, then they have to buy lactate milk, where they've already added this digestive enzyme lactase, so it's already split apart the lactose into glucose and galactose for them, because they can't digest it themselves. And if you've ever tasted lactate milk, it tastes much sweeter than regular milk. We've talked about that before back on D1 when we're dealing with sweetness. So let's summarize this back on F1. On page F1, so on page F1, Roman numeral 2 says enzymes. And we wrote that on F1, each metabolic reaction, what does metabolic mean? Biochemical. Now some of us are thinking, my gosh, Professor Fink, you give us too many words. Well, number one, I can't help the words because that's what this, I said learning biology is like learning a new language. But on the other hand, this word appeared on A3, the third page in your lecture outline. Metabolic. So if you don't know it, I presented it during the first week of class. first couple of weeks. So you haven't kept up with learning stuff that we gave you a long time ago. All right, so anyhow, each enzyme catalyzes a biochemical reaction. Now, how is it possible that, sucrase only attaches to sucrose? Lactase only attaches to, lactase only attaches to lactose. So we commonly say that the way an enzyme attaches to a substrate is similar to the way a key fits into a lock. You can have a bunch of keys in your pocket, and only one of those keys will open your car door, and only one of those keys will open your house. So they're very specific. So, you look at keys, and they kind of all look basically the same, but the specific shape of the key determines which lock it can go into and turn. So similarly, all these enzymes, they just look like, a big mess. But their slight differences in shape are why different enzymes will attach to different substrates. They are very specific based on their shape. So just as a key is specific for fitting into a certain lock, each enzyme is specific for attaching to a particular substrate. So they're very specific. The substrate or reactant is what the enzyme attaches to, acts on. The product is what the reactant or substrate is changed into. And when we name enzymes, we usually take the name of the substrate and just add the ending ase. So if the enzyme acts on sucrose, the name of the enzyme is sucrase. If the enzyme attaches to lactose, the name of the enzyme is lactase. Now, what's listed next? We're almost done as far as what we're asking you to know for the test on Monday. We're going to draw the line right here, so we're almost there. What are coenzymes? What are coenzymes? Now, a coenzyme, you know them better by the names vitamins and minerals. And all of us have heard of vitamins and minerals. We just don't know what they are. Well, actually, you know what? A lot of the words that we've learned in this class, you knew the words already. Just didn't know what it meant. You didn't require this biology class to hear the word protein or carbohydrate or saturated fat or unsaturated fat. You'd heard all those words before. It's just that they didn't, whatever you thought they meant, I don't know if you thought that whatever you thought was correct or not, but what we try to explain is what these words actually mean. So, similarly, vitamins and minerals are coenzymes. Now, I wrote, what are coenzymes? They assist the enzymes in catalyzing biochemical reactions. Here's my analogy. And I've mentioned this before. To fly a small plane just maybe requires a pilot. To fly a larger, more complex plane requires not only a pilot, but a helper, a co-pilot. Maybe even more than one co-pilot. So similarly, to catalyze a simple chemical reaction just requires an enzyme. But to catalyze or make a more complicated chemical reaction occur may require both an enzyme and a helper, a coenzyme. Who are the coenzymes? Vitamins and minerals. That's what vitamins and minerals are. Now, these vitamins and minerals are needed in our diet in small amounts to help the enzymes catalyze these chemical reactions. Incidentally, the original context that I mentioned, this idea of an enzyme and a coenzyme, was actually on page, if I can locate it, was on page D14. If you were to look on page D14, so on the lower left on D14, you will see where I used exactly these words on the lower left. And you'll notice that the enzyme that breaks ATP apart into ADP and phosphate to release energy, the name of that enzyme was called ATPase, because that's how we name enzymes. And then we said you needed a coenzyme or a helper for this called calcium. Calcium is a coenzyme. It helps the enzyme catalyze this chemical reaction. And ATP is really, really important. So finally, back on F1. So on F1, so what is the difference between a vitamin and a mineral? If they're coenzymes, what's the difference? Minerals are inorganic coenzymes. They are inorganic molecules, whereas if we look on F2, towards the bottom of F2, what are vitamins? Organic coenzymes. Now, if we're scratching our heads saying, but what's an inorganic or an organic chemical? We learn that on page C4. Organic chemicals. What are organic chemicals? How did we define an organic molecule, an organic compound, an organic chemical? It's on page C4. It's made up of carbon and hydrogen. It's a hydrocarbon compound. So any chemical that's made up of carbon and hydrogen, it can be made up of other chemicals as well, is called an organic molecule. We learned that on page C4. We need to know that for the first exam. All right? So vitamins are organic molecules. They are large and complex. So on the previous page, F1, then what are minerals? Minerals are inorganic. We learned on page C4, inorganic are just small, simple chemicals. And in fact, most of these minerals are basically simple atoms. They're just simple atoms. That's what minerals are. So in fact, an example of a mineral is calcium. Calcium's on our periodic chart. Right? It's just a basic, it's a metal, it's a metallic atom, a metal. Gives away electrons. So, so that's what minerals are, it's just these simple, it's not an organic molecule, it's just a simple metal or non-metal. So those are minerals. So I'm not going to include this on the exam, but what we're about to start to talk about are minerals and vitamins and what they do.